Hello, good morning, good afternoon, or good evening, wherever you are. My name is Muthukrishnan Renji Ranganathan. The Renji is the nickname for me. I'm the Senior Study Director at Eurofins Discovery, St. Charles, Missouri. I've been with the company for the last 14 years and mainly focusing on cardiac safety. And actually, cardiac safety I've been working on for the last 20 years. So I have seen the technological evolution of this area. And the last 2 years, I've been also, our company also been focusing on the vascular safety for the drug discovery. So that's all is about, I'm going to be talking about for the rest of the hour. That's the title is the comprehensive identification of cardiovascular safety, liabilities using a totality of evidence approach. So in the next slide, the outline for that my talk is given here. As I mentioned in the title, my talk is twofold. One is to talk about the cardiac safety evolution for the last 15-20 years. And the next, the second part, I will be talking about what we are doing in Eurofins to help the customers for the vascular safety. You know, just like the cardiac safety, even the vascular safety, some of the drugs were withdrawn. And I have some, background issues I can talk you through. So basically, for the cardiac safety, what we wanted to say, talk new here is the FDA has come up with the in vitro best practices for cardiac safety evaluation last year, and then I will probably give us some background on drugs withdrawn due to torsade or arrhythmia, cardiac arrhythmia, and then for the last 20 years. What are the major initiatives of non-clinical cardiac safety evaluation that was done, to streamline the cardiac safety, and the challenge of predicting cardiac safety and the problems associated with it initiated the new paradigm called the comprehensive in vitro pro-arrhythmia assay, and that we've been working with the FDA and HESI for the last 5-7 years. And we also have a new high-throughput platform called Cube 384 that is a game changer in high-throughput ion channel screening and how it ties into the in vitro best practices for cardiac safety evaluation. That was listed in ICS E14 S7B and S7B Q&A 2. I will explain that. And then how we validated the 3 core channels, cardiac channels, sodium 1.5 peak current, calcium 1.2, and the hair repolarization current at physiological temperature, and why do we have to do at physiological temperature. And I'll also show you what we've been doing for all the 7 channels listed in the comprehensive in vitro pro-arrhythmia assay. And I also talk about briefly about what we are doing with the iPSC-derived cardiomyocyte with Micro-Electrode-Array to help with the cardiac safety evaluation. And then I switched to vascular safety with our biomarker assays, because as in cardiac safety, the vascular safety, the drugs were withdrawn to vascular and bleeding issues. And HESI and FDA, they are the one who usually work together, to form a working group and coordinate all the things, just like what we have done for cardiac safety. And I looked at the HESI vascular safety working group and what are they thinking, how they want to address this vascular safety, because the drugs that were withdrawn, Vioxx and Bextra were 2004, 2005, and a few other drugs in the 2010s. What is happening, what they are thinking, I will talk about a little bit. And then how we tie that into our biomap platform, where the 3 key pillars can help us to evaluate the vascular safety. So I will talk about how our biomap systems incorporate physiological relevance and significance, and specifically the biomarker assays thrombosis for bleeding issues and strokes and vascular toxicity, with some other cells. And then finally, I'll show some data with, the drugs that were withdrawn, Vioxx and Bextra, and celecoxib was the black box warning as positive controls in the biomarker assay. That's going to be my outline for the talk. So this figure shows the totality of evidence approach for in vitro, both the cardiac and vascular safety, by looking at the high throughput ion channels screening together with the translational models such as the biomap platform that will provide a comprehensive assessment. And you can see on the left side the cardiac ion channel assay, there are 4 buckets. You can see we have given the herd channel block, but we are looking at all cardiac important channels, and then using these in vitro data to model and predict, in silico model, what's happening to the cardiac action potential. If it is prolonged, a sustained prolongation, can you even see EAD? And that's then checked, verified with either cardiomyocytes, and to see the prolonged QT interval or EAD, which normally is seen in human clinical study, very rare, and that's the Torsades-DuPont. And that's the basic idea. We're looking at 2 key primary endpoints: the ion channels and the QT prolongation, because the Torsades-DuPont is very rare. These are surrogate biomarkers, the QT interval and the herd channel block. Similarly, we are going to I will show the data for biomap human phenotypic platform, thrombosis and vascular toxicity, how we can help with some drugs that were withdrawn, some that showed black box warning. So this is the slide that shows the major initiatives of non-clinical cardiac safety evaluation for the last 24, 25 years. And starting from pre-1999, there were some key drugs were withdrawn. These drugs went through the non-clinical and clinical safety evaluation and went into the market, for a year. And then the patients showed some kind of arrhythmia and Torsades-DuPont, and they had to be withdrawn. So all the stakeholders, like the FDA, regulatory agents, and other regulatory agencies of the world, and the big pharma and CROs like and academics, they all came together and formed these guidelines called ICH S7B and G14. The S7B is for the non-clinical guideline, E14 is for the clinical. And we were the first one around 2005 to start the 96-well plate throughput in Ironworks Quattro. And that time we screened cardiac 8 when we used a perforated PET clamp. And that's like old technology, but I don't want to go in details, but we were the first one to do it. So what does it do? These surrogate endpoints, human UTHR-GOGO related gene herd block and the other 7 channels, and also evaluate QT prolongation by looking at the cardiac repolarization assay. These are very highly sensitive, but not very specific for predicting proarrhythmic risk. So, and there was some feedback from the pharma, that to the FDA that we're having some issues with this kind of guidelines. So around 2013, all of them again got together and then evaluated the data they had, and then the new paradigm came for assessment of clinical Torsades-DuPont. This is the PCiPA cardiac in vitro proarrhythmia assay that is not measured exclusively by the potency of herd block. So, and irrespective of QT prolongation. I will explain what is PCiPA briefly and how we are helping the customers. And as I said, we've been working with FDA and HSE for the last 5-7 years, and it's evolving, these guidelines. Even the PCiPA paradigm is evolving. And one of the key aspects was during this evaluation, what the organizer found was the clinicians were not looking at the non-clinical data, because the ion channel data, most of them were done around room temperature, or even when they're done at physiological temperature. The protocols to test these ion channels were highly variable. Each one had its own protocol, and it's not a cohesive information FDA can rely on. So that's why they kind of put us all together to put together harmonized protocols for the cardiac ion channel safety evaluation, and also to do all the 7 channels at physiological temperature, that is at 37 degrees Celsius. Now I'll show you how Cube ties into this, how we are able to assay all the 7 channels at physiological temperature for a totality of evidence approach to better understand drugs effects and the consequences in the clinical setting, that is in a physiological temperature. That's the main focus of the part of this talk. So this table is from Norman Stockbridge, who is the divisional director. He's been there for almost 20 years. And he's the one who summarized all the data from 2003 to 2013. the title is dealing with the global safety issues. Was the response to QT liability of non-cardiac drugs well coordinated? And the title is given, and then the link to the publication also listed here. And there are 3 columns. Column 1 on the left extreme is the name of the drug, and the second column is the therapeutic class and the year withdrawn. And you can see most of the drugs were withdrawn around, from 1990 to 2003. After 2003, introduction of the guidelines, very few drugs were withdrawn. And you can also notice the top 2 drugs are the anginal, that means the cardiac drugs. The rest of them were non-cardiac. So that was the big issue, the drugs were non-cardiac, but they have a cardiac effect. So that's the spookiness of this nature of this, the HERG issue and other issues. So, so when they looked at all this data, they came up together. So HERG only focus, is good. It cut down the drug withdrawal, but it came with some other issues. And that's shown in the next slide, that the new paradigm for cardiac safety evaluation from 2013 onward. So all QT prolongation is not pro-arrhythmia. All HERG inhibition is not pro-arrhythmia. There was evidence in the literature, even before the guidelines were introduced in 2003. Fluoxetine, the Prozac, is an antidepressant. It's a multi-billion-dollar drug, and it's safe. And verapamil is also an anti-arrhythmic agent and safe, even though they block the HERG current potently, but they also block the calcium 1.2, the L-type calcium current at the same concentration under cardiac safety. Fluoxetine was developed in the 80s, and verapamil was developed in the 60s, even before we know anything about HERG channel. And that's how it's kept. If it was introduced in 2003 or 4, if they see the HERG block, this would have been a cutting line. They will be in the cutting area. So, so this kind of in the background was the information the big pharma was telling FDA. Look, we cannot simply focus on HERG block. And then the other drug is ranolazine, also a HERG blocker, but also blocks sodium late current at the same concentration in cardiac safe. So we can take the HERG only focuses negative impact on drug development. You may lose good drugs. And the new paradigm for cardiac safety evaluation comprehensive immunotherapy arrhythmia assay was introduced in 2013. The main idea is do not miss out on the good drugs by HERG signal, but at the same time decrease the post-approval marker withdrawals as shown in the bottom figure. So in the right panel figure, there are 2 panels, one on the left side, one on the right side, and you can see A and D represent the cell membrane figure, and then in the B and E is the cell type from a heart, and then C and F from the ECG of the human body. So the left-hand panel shows the data for predominantly HERG block that leads to the Torsades-DuPont. As we see in panel, this panel A, the HERG block is blocked. The HERG current shows the block of the potassium current flowing outside, and you can see the sodium and calcium current open. So what happens when you block only the potassium current is the repolarization that happens because the outflow of potassium gets delayed. And during sustained delay in repolarization, the activation of a sodium late current or calcium current in the plateau phase can induce early depolarization, early after depolarization. That can degenerate under very rare circumstances to Torsades-DuPont at the body the body level, and that's the issue that makes the drugs withdraw. But on the other hand, if you have a balanced ion channel block causing QT prolongation with the Torsades-DuPont, the main difference here is that in addition to the HERG block, you also see block of the calcium channel and the block of the sodium late current. So what happens is you still see a cardiac repolarization delay at the heart cellular level, but the delay is not detrimental. Even if you look at the tissue level, body level, that the delay is there, but it does not degenerate into Torsades-DuPont. So the main take-home point from this slide is, if you have a HERG block, but also at the same time, if you have either the calcium block or the late current block, your drug is safe, cardiac safe. You don't have to cut it. That's the main, the take-home message from this slide that was published in 2018 in the CPT journal.com. So that initiated the comprehensive in vitro pro-arrhythmia assay proposal that's described in this slide, and it has 4 buckets, 1, 2, 3, and 4. Bucket number 1 is to in vitro assessment of drug effects in multiple ionic currents that I'll talk about later, the CIPA7, mainly the sodium and calcium, and several types of potassium currents. And once you get the IC50 values or the inhibitory values, you take these values, integrate the ion channel effect in silico computed modeling to predict risk. There are 2 portals available, one in the FDA, one in Oxford. These portals can allow you to put input to the data and then predict what's going to happen to the cardiac action potential. At the bottom, it says predict clinical risk of arrhythmias, whether you was going to see a sustained delay in repolarization or EAD. And then that can be checked and confirmed in vitro effects on human stem cell-derived ventricular cardiomyocytes. Why do you want to do that? The cardiomyocytes have all these ion channels in them. In addition, they also have the exchanges, sodium-calcium exchanges, other transporters, and an adrenaline, noradrenaline affecting system in there. So in other than these ion channel effect, any other effect on the cardiac functioning can be found out in these cardiomyocytes, just before you go into the phase 1 clinical trial. In phase 1 clinical trial, you have the whole other systems interacting with you, CYP inhibition, liver toxicity, and other stuff. All these things can be, evaluated quickly on that. And also, if bucket 1 to 3 is so clear, then the phase 1 also shows very clean data. Then there is no need to go for thorough QT studies, which clinicians, when the FDA suggested this thorough QT, that was the most time-consuming and expensive study the big pharma were very hesitant to do. And so this is kind of a streamline and clearly tell with a scientific approach whether you have to do the thorough QT study or not. And so in a way, the CIPA consortium, CIPA-HC and FDA consortium, what did it do? It harmonized ion channel assays. Just like us, there were several stakeholders that we all came together and harmonized the voltage protocol. It took a year because of the consensus, but finally we got it done. And that enabled regulators to review cardiac safety study results more quickly because, they're all coming with the same voltage protocol across submissions, okay? And then we also participated as a test site for the CIPA-28 compounds. These are a group of compounds that were either withdrawn or medium cardiac safety issues or low risk, and that were blindly tested on us and to check, mainly for the cross-site and cross-platform variability of automated path climb assessment of drug effects on human cardiac currents and recombinant cells. This paper came out in 2020, and another, the modeling paper came out a little bit before, also in 2020. The main aim of this is, these 2 papers suggest that there's not much cross-site or cross-platform variability, and this can be used to evaluate the cardiac safety, and mainly to integrate the risk assessment of both non-clinical and clinical data to provide a totality of evidence approach between the non-clinical and the clinical data. And inform late-phase clinical trial design and labeling for market application that come from both from the non-clinical and clinical data. Till 5 years back, clinicians never looked at the non-clinical data, but now there is an emphasis. Basic science and clinical science all has to be taken together to evaluate the risk. That's to mainly reduce the number of thorough QT corrected clinical studies to reduce cost and improve safety. Now, here in this slide, I'm talking about the platform Cube 384 high throughput screening system from Sophion Bioscience. That's the picture given here. And as I mentioned earlier, I've been in this field for the last 20 years, and I have seen the patch express with the 16 well platform, and then the cube patch as a 48 well, and quattro again 384, and cube 384 as a 384 well platform. The main difference between quattro and cube is quattro, it is not a continuous patch clamp, and because of the uniqueness of the system, whereas here the cube is a continuous, just like the gold standard of the manual patch clamp, a continuous evaluation of the ion channel currents. The 384 amplifiers provide individual adaptive patch clamp recording conditions to ensure tighter data distribution with reliable, more predictive results. On top of that, the assay temperature can be set at any desired level and kept constant. In our hand, we wanted to keep it at 37 degrees Celsius ±1 at physiological temperature. Thereby, the Cube 384 offers complex ion channel integration with high fidelity recordings, accurate liquid handling, unattended operation, robust statistical power at competitive price. So this is a game changer for all the high throughput ion channel screening assay, and we are very happy with it. Now, the in vitro best practice that came out in 2022, based on the clinician and non-clinician discussion, it came up as a question and answer session for ICH E14 S7B or S7B Q&A 2.1. Q&A 2.1, where it talks about the recording temperature. The recording temperature impact on herd current has been reported for several molecules, including erythromycin and most of the antibiotics. Their IC50 value, the inhibitory concentration at 50%, is 7-fold higher at ambient temperature than at physiological temperature. That came out when I was in Chantest in 2004, and one of my colleagues at Chantest, that came out 2005, and he moved to East Coast to work with another big pharma. And there were other publications that came that elevated recording temperature can lead to both increases and decreases in herd, both increases as well as decreases. That's the point. In herd IC50 values in a test article specific manner that came out in 2006 and 2015 with the other two publications. And on top of that, there is no method to predict physiological temperature IC50 values based on room temperature results. Therefore, the in vitro best practice, in addition to all other guideline suggestions, they recommended recording temperature to be at or near physiological temperature in order to better predict the test article's impact on ion channels in the clinical setting. And one point I want to mention is that most of the studies have come up only for the herd. We don't know nothing about the other 6 or 7 channels, what kind of compounds or temperature dependency. So, and how this can play a big impact in the evolution, supply evaluation, I will summarize in a few slides later. So now with the availability of the Cube platform that can assay at a physiological temperature, we started validating the now 1.5 peak current assay using the PCiPA harmonized protocol. That is the red line shown here and at the left panel. And we are holding at -80 and step down to -120 to recover from inactivation. And then you depolarize to +40 millivolt, but on the step, you are stopping at -15 because that's where the sodium peak currents activate maximally to measure the peak current as the second green line, green bar. This, the first green bar is the measures the leak current. And during this plateau phase, there is no leak current, and even during the ramp phase, that is to mimic cardiac action potential from +40 to -80 millivolt. That's the repolarization, and the going from -120 to +40 is the depolarization, mimicking cardiac action potential. And you can see the current is very stable and very robust and very, very stable. And that's what is shown on the right panel, the IT plot of the vehicle control of the peak current and the leak current measured here, and the temperature of each well that is monitoring this current. So here is the sodium peak current 5 minutes after breaking into whole cell, and then 2.5 minutes of vehicle control before applying a positive control of tetracaine 1 millimolar 2 times, for a total duration of 5 minutes. Normally, we expose the compound for a total duration of 5 minutes. And you can see clearly the window of the assay from, from 15 nanoampere to 0. So very good window, very stable. And we were a little bit, just like any other electrophysiologist, we wanted to know how fast this clamping of sodium peak current will be affected by the 37 degrees, because it speeds up the process of channel opening, but it looks like very stable, as we see here in the stable baseline. And in the next slide, I'm showing the compound from the CYPA28 compounds. I selected 6 compounds that show some kind of sensitivity physiological temperature. So, for example, clozapine, there is a 10-fold shift, from 23 to 31 micromolar at ambient temperature. This is the estimated IC50 value with the 95% confidence interval of that IC50 micromolar, and you can see the dotted line next to each, stable, non-dotted line, and that is the 95% confidence interval. And you can see there is a 10-fold shift for clozapine. Similarly, droperidol, loratadine, pimozide, diltiazem, and pepridol, also some kind of, assay temperature dependency. So in our hand, diltiazem and clozapine is the most temperature dependent, and these values are either close to the manual patch clamp value or a little bit poorer than the manual patch clamp value. And here we are showing the calcium 1.2 peak current assay at physiological temperature using the CYPA protocol. Again, the red line here on the left panel. So we are starting from -80 millimolar holding potential, stepping to -100, and then we step to +40 after a brief holding at -80 millivolt. On this depolarization part, we are stopping at 0 millivolt for briefly about 30-40 millivolts to elicit the calcium peak current, because that's where they activate maximally, and you can see this here. And then we step to the +40 millivolt to mimic the Claude phase of the cardiac action potential, and that is shows that a slow inactivation. The currents are inactivating. From here, they inactivate and then they keep inactivating. And then during the repolarization assay, all these channels that are not inactivated, they open again. So for electrophysiologists who are in this talk, they can understand why we are measuring the peak current and the ramp current. The peak current is to measure what's happening when the channel open, and the ramp current to show if there is any state-dependent inhibition due to inactivated state, and that's what we said here. But here I'm showing only the peak current measurement and the leak current measurement and the temperature that was monitored. So here is the 2 additions of vehicle control, and then again another 2 additions of vehicle control, total of 3 minutes, and then we add nifedipine for about another 3 to 5 minutes. I'm sorry, this is 3 to 5 minutes, and you can see that nifedipine blocks almost 80% of this current, 80 to 90% of the current here, but at the ramp it is completely inhibited, due to the state-dependent inhibition. And you can see in the right axis shows the temperature values, right y-axis, and that's the triangle is the temperature being monitored throughout the measurement and kept at 37 degrees Celsius. So these are the 6 compounds again that show some of the sensitivity to the temperature. Clozapine, again with the 95% confidence interval, micromolar, IC50 micromolar for ambient and physiologic temperature. We have the clozapine, domperidone, metoprolol. They don't show much of an effect, but you can see nifedipine and pimozide, they show temperature dependence, and the nifedipine shows a little bit, not much effect at all. So, but we have all the compound CYPA28 compound data. If anyone is interested, you can reach out and we can see how we can communicate that with you. And here comes the Herg assay at physiological temperature that was again monitored at the super harmonized protocol, holding at -80 before stepping to +40 for about half a second, just mimicking the cardiac action potential duration of the human cardiac beat, and then repolarized back to -80 millivolts. So you can see that the Hurd current, they activate and inactivate faster, and is, you know, in the plateau phase, because that's their silent characteristic. And then when they step back to repolarization state, all the inactivated channels open, and they inactivate very slowly. That's why you can see a huge current, what we call the tail current, and that's measured just like what you see in the cardiac tissue, you know. And this peak current is measured as the tail current, and the lead current at the holding at -80, and that's shown here 5 minutes after break into whole cell, 2.5 minutes, and then another 2.5 minutes addition of E4031. And then you can see also the temperature that was monitored during this experiment listed at the triangle, and the axis given on the right hand side, y-axis. Then why did we add 2 additions of compounds? Because we don't know what your compound is going to be. It's going to be highly soluble in water, or it could be very sticky. And if it is sticky because of the setup of the cube, what we wanted to do was we add the compound twice. You know, first for 2.5 minutes at the same concentration, we replace that compound again with the second fresh addition of the same concentration. And here the example is a E4031. This is a very highly sticky compound, and you can see the second addition induces more inhibition. This is mainly to, you know, address any sticky compounds which we are not aware of, you know, from your compounds that are sent to us for screening. So, and again, there are 6 compounds I picked up for showing the temperature effect, and you can see diisopropamide, butylide, dofetilide, tefenadine. They also some kind of temperature dependence. These, you know, look at dofetilide and tefenadine at 60 nanomolar, 70 nanomolar, and very close to what we see in some of the Benjamin Pachkam values. And the main interesting aspect is both the sotalol and clarithromycin shows a very high dependence on temperature, and particularly clarithromycin shows about 10-fold difference, and sotalol shows about 5 to 7-fold. And these are the values, you know, the differences the FDA wanted to see, you know, and other people wanted to see. And here I'm showing some of the other CIPA 7 channels. I have already talked about the 3. This is the 4th one, CIPA KV 4.3 chip 2. Again, you can see the red traces of the harmonized protocol, and we are showing what we did with the vehicle control, you know, the time, you know, IT plot, and the 2 vehicle control, and 2 addition of flecainide, and then here the IC50 value of 3 micromolar flecainide, and then 2 additions of 300 microflecainide. So these are all data being collected, and we are analyzing the data, and then we will probably be available, you know, end of the month or end of September. Similarly, the late current antagonistic assay. You know, remember we talked about in the early slides, the late current block is very safe or mitigate the effect of the Hurd current. So we need to measure this at physiologic temperature that we induce with the ATX2, and you can see the peak currents very slow to inactivate, you know, and you can also see the ramp current. Unlike in the peak current, sodium current, you can see the ramp current because it's the late current. And again, we use the same CIPA harmonized voltage protocol, and this is the IT plot vehicle control of the ramp current and the lead current and the temperature. And this is the ramp current. This is after applying the whole cell configuration, and then we add the ATX2, and then we add ATX2 again, and then we add ranolazine at high concentration to block it completely. So here comes the KIR 2.1 assay at physiological temperature. Again, we mimic close to cardiac, you know, the harmonized assay protocol, and we have the vehicle control data for DMSO. You know, if you add DMSO, there is some not much effect in here. This is 5 minutes after vehicle obtaining whole cell configuration and then vehicle control, and adding the 100 micromolar barium chloride as a positive control to block the current. And here we are showing that what happens if you use a 10 micromolar barium chloride. That's around IC50 value. You can see 10 micromolar blocks about 60-70% of the current, and that's what is the data shown here. And we are yet to collect the data for KCNQ, which is in the works. And once it is done, we will be able to offer all CIPA-7 in the late September or in the middle of September. Here, I wanted to emphasize the comprehensive in vitro arrhythmia assay. What is the why we should be doing the physiological temperature? If you look at it, bucket 3 and 4, they are done in physiological temperature. Remember, I mentioned 5 years back, most of the people used to do this assay at room temperature. And even if you do the computer modeling, if these IC50 values become more potent at physiological temperature, then all bets are off, right? You are trying to predict and then go and verify, then there'll be no cohesiveness. You may have a surprise. This is why it is important to do all the 7 cardiac channel at physiological temperature to keep it same for all the 4 buckets, the assay conditions the same. That's the idea of the in vitro best practices. And that's what we're trying to do and help the customers. And we could be the first one offering all the 7 at physiological temperature. Now I'm showing the Micro-Electrode-Array with the cardiomyocytes using specific positive control, like for sodium channel blocker, mexiletine, diltiazem for calcium channel blocker, and U4031 for hERG channel blocker. And here you can see there are 2 lines in here. The first line is the depolarization, and the second peak is the repolarization peak. And this is the the equivalent of QT prolongation, but in the cardiomyocyte assays is called field potential duration. And when you add the sodium channel blocker, that elicits the depolarization peak. It is blocked, and you can see it reduced more than 50 to 80%. But there is no effect on the field potential duration. On the other hand, the calcium channel blocker, diltiazem, I talked about the second peak is the field potential duration measurement, between these 2 peaks. And when you add calcium channels, that blocks the plateau phase, it reduces the plateau phase, and the field potential duration decreases. On the other hand, U4031, it delays the repolarization. So what happens? The repolarization peak moves away from the depolarization, and that increases the field potential duration. And that's what is summarized here for the control and then the compound addition for the sodium channel block, a decrease in spike amplitude, a calcium channel block, and the field potential duration decrease, and hERG channel block follow the button for the increase in field potential duration. This is from one single well. But if you ask, how does a scatter plot for MEA parameters look? Then this is the scatter plot with the 90% confidence interval with the line, a dotted line and small dotted line. This is the 90% confidence interval. So you can see the 30 nanomolar E4031. You can see there is a increase in the baseline due to the increase in field potential duration and the corrected duration, but no effect on the spike amplitude. And similarly, the mexiletine only blocks the spike amplitude, so no effect on beat period, field potential duration, or Fredericia correction of the field potential duration, but an effect on the spike amplitude. And diltiazem, as I showed, that blocks the calcium channel, shortens the field potential duration, and you can see the beat period decreases, field potential duration decreases, as well as the corrected one, but no effect on spike amplitude. And this is all control, evaluated in the presence of vehicle control data. Just like if you add, normal compound media that is used for the compound. In this case, only the media, no compounds, and you can see there is no effect, and then it falls. That's determining the 90% confidence interval. Again, so we are trying to help the customer with the 2 main buckets of the in vitro assay, and the second bucket can be done in the FDA portal or the University of Oxford portal before they go into the phase 1 clinical trial. Now I'm switching to the second part of the talk, that is the drugs that were withdrawn or had warnings added for potential vascularisis. Viax is a non-steroidal anti-inflammatory drug, very popular around 2000. The same as the Bextra, again, another NSAID drug. Both were withdrawn in 2004 and 2005 due to increased risk of cardiovascular events, in the first case, a heart attack and stroke. Celebrex, that was introduced by Pfizer, was required to add a black box warning stating the potential for increased risk of cardiovascular events, life-threatening gastrointestinal bleeding. We all know how much time and money it takes to introduce a drug to the market. It takes like 15 years. Around 2000, it was probably 1.1 or 1.5 billion dollars, and so all the investment and time is wasted. And 3 drugs were withdrawn. And one of them had a black box warning, reducing the sales figures, particularly increase cardiovascular events and life-threatening gastrointestinal bleeding for Celebrex. And it also happened for the antiplatelet drug Brilinta, and then anticoagulant drug Pradaxa, and then cholesterol-lowering drug Zetia. These are all had a warning ordered for the around 2011 and 2020. So this is not a thing Big Pharma wants to see. So how can we help them? How can we help them just like the way the cardiac safety has helped the customers? And that's what the FDA and the assay has been working on. So I've been looking at their website from 2019 onwards, and then when you look at it, the assay cardiovascular safety. Remember, they're talking about cardiac and vascular safety together. Subcommittee initiative that was evaluation of 3 non-clinical models of QT prolongation. That was introduced as soon as the 2 drugs were withdrawn around 2004 and 2005, around 2006. And around 2010, Dusty Zarazan and other people, they formed the working group for cardiovascular function in non-clinical drug safety assessment, current issues and opportunities. Basically, how can it be make it better? And then they call for more integrated cardiovascular assessment in 2011. And around 2018, I don't know what happened, after 2011 because of the introduction of the cardiac safety. They were waiting for the resolution of the cardiac before they focus on the vascular safety. Around 2018, they formed another working group specifically looking at preclinical cardiac biomarkers of hypercoagulability induced under thrombotic state in both normal and disease state. And how they were going to do was to investigate the effects of doxorubicin in Zucker diabetic fatty rats. And they were in the planning stages of using xenobiotics, that is a drug discovery, to induce the precoagulant state and confirm measurements of biomarkers of interest. But the FDA Modernization Act 2 that came in 2020 authorizes the use of certain alternatives to animal testing, including cell-based assays and computer models, basically to obtain an exemption from the FDA to investigate the safety and effectiveness of a drug. Basically, what they're trying to do, we know non-clinical, fails some of the compounds because they are withdrawn. And, they are focusing on, what can we do to better introduce in vitro assays that most resembles the human tissue system. So that way we don't get any surprises as a drug being drug withdrawn. That's the basic idea to help the drug discovery. And that's where our Eurofins in vitro cardiac vascular safety assay comes in. Here I'm talking mainly about the translational models such as the BioMap platform that provides a comprehensive assessment for the vascular safety by looking at the thrombosis and vascular toxicities. So the next slide, I will talk about what we are doing at Eurofins about the BioMap phenotypic platform. The BioMap phenotypic platform uses integrated systems biology and advanced analytics for translated insights by using 3 key pillars. Pillar number 1 is the different types of human primary cell models and readouts. That is the BioMap system. So the human primary cells and co-culture, broad coverage of tissue and tissue biology, and clinical biomarker. These are the protein endpoints. So thereby they provide the highest level of physiological relevance. And we have a reference database of more than 4,500 agents that are drugs and clinical stage, approved, failed, and withdrawn, and experimental compounds, tools, and mixtures. And we even have used the biologics, antibodies, proteins, peptides. So we have a unparalleled level of validation and guidance to look at, how your drug is, can be used, can be evaluated with our reference database. And in the third pillar, we're using the predictive analytical tools, the mechanism of action insights based on our data, and toxicity signatures, and then combination analysis. And then we can do the benchmark against approved drugs. And that's the idea of the reference database and the predictive analytical tools. Just like FDA recommends sometimes, you have to benchmark against a drug that was withdrawn from the market. So that way we are already prepared to help you in all of these assays by having this big database. So the biomap systems incorporate physiological relevance. To emphasize again, the human primary cells has the highest physiological relevance, and we also do the co-cultures. Like for example, if you want to do the effect on the cardiovascular, we have the cardiac tissue and then vascular effect from endothelial cells and the other tissues from aorta and all those other things that can be co-cultured just to mimic what's happening in the tissue level, to capture cell-to-cell communication and the tissue level. And then the assay conditions, the combinations of stimuli, we can do that to induce more representative of a tissue state or a disease state. And then we can monitor the clinical protein biomarkers, that can, facilitates clinical translation. So the biomap standard services, we are offering a bunch of services, but I just want to quickly go through. We are offering to start with the profiling panel. That's a diversity plus panel. I don't want to describe everything, but it is, the mechanism of action, everything is listed in here. The T-cell autoimmune panel, fibrosis panel, immuno-oncology, TME panels. This is to evaluate your compound's effect, what is it doing at each level, and what we can tell, as due diligence or benchmarking. And the next point is the diversity early screening or systems early screening, and then profiling screening, system elect, you can select, and then the combo elect, you can tell, if your compound is hitting two different systems of tissues, we can use that, and then the RNA elect. And then the analytics uses the toxicity signature analysis, and we give a data packages and overlay elect, and we help you through with all the data by explaining and working together to write the report for you. And we are very good in customer service, and our goal is to make it very easy for the customer. Now, here is the biomap diversity plus panel. It has a 12 assay system, 148 total endpoints to capture broad range of human biology. And that is the vascular system, the 4 assays, and then the fibrosis system, and these are the another 4 assays, and then the immune system that uses another 4 assays, and then we have the respiratory system. So that way we can monitor these 4 key area with the 12 assay systems by looking at the cell proliferation, DNA repair, and vascular biology, immune biology, and stimulation conditions model adaptive responses. And we can look at inflammation, immune response, tissue remodeling, wound healing. These are some of the issues that can become, initiators of the vascular issues, and something like that for other, neuronal and other issues too. So the toxicity signature analysis, as we talked about, we are using these 12 systems, and then we have the 9, toxicity assays from acute to thrombosis and vascular toxicity, as well as liver, skin rash, and immunosuppression, and organ toxicity. But here in this case, we are talking about the thrombosis as a highlight. Here we are defining the increased incidence of stroke, deep vein thrombosis, and pulmonary embolism. These are very serious issues for human health, particularly around 50s and 60s. And we have key reference compounds for this thrombosis, and these are mentioned here. And we use different classes of drugs. to just to see what kind of, just to mimic or just to represent the different areas of drug discovery. And we have the biomap key biomarker. This is listed here, biomap C3 system, 3C system, and pathways and targets listed here. And we also clinical mechanism, modulate vascular autophagy, leading to increased thrombotic potential within the vasculature. This we recommend to do, at the late stage. Flag candidates with the pharmacology signature related to toxicity mechanisms, and to prioritize the safer candidates if you have a handful of them for increased clinical success in the clinical trial. So the comprehensive evaluation of compounds for clinically relevant biomarker activities, to summarize, what we are doing is we are profiling your compound against a set of biomarkers, and we benchmark against the known compounds, from our data points, or if you want to check with your known compound that FDA recommends, we should have it. But if you don't have it, we can use your compounds. And then the top similarity match of the results. What are the things that is lighting up? And then gives a database matches and the mechanism of the heat map, and then the similarity network. So in a nutshell, we give all of this to evaluate, a totality of evidence approach. What are the key biomarkers that is lighting up, and where you have to focus your attention. And here is the explanation of diversity plus profile. So here, again, as I mentioned earlier, 114 protein-based readouts, biomarkers, and quantitative protein readouts, either soluble or cell-associated, and proliferation, cytotoxicity, readouts for therapeutic and biological relevance, and validated with well-characterized therapeutic agents and mechanistic toll compounds. We are also evaluating the literature as in every field, as the technology science evolves, there are more information of biomarkers, and as they are coming in, we are also introducing into our assays. So we are mostly trying to be updated or upfront or more current in our assays. So what we are measuring is the relative expression levels in a log scale of post over pre drug application over vehicle control. And we can apply the drug, either 24 hours, 48 hours, 72 hours, depending on how you want to, apply, evaluate your drug. We can do that. We can work with you. And we are, using these systems at the top. What are the things that are happening? And we can detect cytotoxicity, anti-proliferative effects, and then we are looking at the annotated effects, that the markers that are showing effects due to the drug exposure. In this case, we are talking about IP10, and we can also give the 90% confidence interval of historical controls. So you can have a high confidence in your data of what these compounds are doing and what you take from this data. And right now, so I'm switching to the as we talked about in the outline. I'm using the y-axis as the drug. We evaluated at 4 concentrations: 100 nanomolar, 1 micromolar, 10, and 100 micromolar. And you can see some of these biomarkers lit up. This is the prostaglandin E2 soluble, and the same as the tumor necrosis factor alpha, that's also soluble form. And you can see they are decreased, but there is an increase of vascular cell adhesive molecule 1, and then tissue factor 1 also decreased. And then IL-10 also is also being seen. So the take-home message is they are active in 5 annotated readouts. The cytotoxic concentration, no cytotoxic activity, and no antiproliferative effects. And the main effects are seen in a decreased tumor necrosis alpha, SPGE2, VCAM1, and decreased interleukin 10, soluble interleukin 10, and decreased tissue factor. And I will talk about some of the key biomarkers on the last slide, because I don't want to take him explaining what is going on. The same as the Baxtra, that it is, again, the same concentration range. There were 10 annotated readouts, some of the key being NCP1 and TM, and then CD38 and collagen 3 and CD69, in addition to interleukin 10, tumor necrosis factor alpha, and PGE2. So this explains whatever effect, antiproliferative effect, inflammation, or immunomodulatory, tissue remodeling, hemostasis, and all this is summarized here. Similarly, for Celebrex, we tried 4 concentration, the same as the other 2, but unfortunately 100 micromolar was excluded from analysis because it precipitated. And the Celebrex is active with 6 annotated results. It is not cytotoxic. It is anti-proliferative to human primary fibroblast, plasminogen active inhibitor 1. And then they have the same inflammation-related activities, immunomodulatory activities, tissue remodeling activities, as well as hemostasis-related activities at TF. So these are the biomarkers that are activated by Y-AX, Bextra, and Celebrex. I don't want to go into the detail, but in the slide, when it can be shared, you can read through them. Most of them, light up, with the coronary artery disease or peripheral artery disease, or anti-inflammatory cytokine, or vascular inflammation, and again, vascular endothelium affection. So those are the key points that came up with. And for thrombosis, you can see plasminogen activated inhibitor 1, and then alpha smooth muscle actin. These are some of the key factors that are biomarkers that have been affected in this assay. So, to summarize, at a totality of evidence approach, in one assay at the same site, we are going to be monitoring in vitro cardiovascular safety for the cardiac ion channel using the PCiPA-validated 7 ion channels and the cardiovascular myocytes, and then in the biomap human phenotype platform, thrombosis and vascular toxicity. So you can, just before the in vitro, first in human studies, you can have a great faith and confidence in your compounds. And these are the totality of evidence-based assays for in vitro cardiovascular safety. We talked about the core NaV1.5, calcium 1.2, the HERG at physiological assay, either at 1 or 6 concentration, the assay temperature listed here. As I said, the other sub-4 are being validated. So currently we are offering the Super7 full panel at ambient temperature, but it is going to be available by the 4th quarter. The full 7 panel will be available for the 4th quarter of 2023 at physiologic temperature. We also offer the cardiomyocyte assay to verify the integrated effects of the cardiac channels effect verified, and any other surprise is not there. And similarly, we are going to be we offer the Biomap Diversity Plus alone, or Biomap Diversity Plus with the toxicity signature analysis at physiological temperature. And these are the catalog values. So you can reach out to our BDs or our website to look at these aspects and how we can help you in your drug discovery. And thank you for listening. And you can reach out to either Justin Lipner, who's the Biomap key point, and I am Ranji Ranganathan, the key point for the ion channel screening. Thank you for listening.